Welcome, class, to today's lecture over viruses and bacteria. And today I'm going to lecture over the first part of our PowerPoint, our note outline, which is focusing on viruses today. And it's pretty appropriate considering that right now we're in a COVID-19 pandemic, which is a virus. So today, let's go ahead and start our conversation over viruses. And guys, the first thing that we need to discuss when it comes to viruses is the controversy that kind of comes with them. Are viruses living organisms? Well, when it comes to viruses, the scientific community is pretty much split when it comes to are they living or are they non-living? It's pretty much like a 60-40 split. And the reason there's controversy of if they're living or not is when we look at the characteristics of life. For an organism to be considered living, they have to kind of check these boxes. They have to be able to reproduce on their own. They have to be able to metabolize on their own. They have to be made of cells. They have to grow and develop. And when we apply those to viruses, they don't check off all these boxes. First of all, they can't reproduce on their own. They could only reproduce when they attack a cell, when they get a host cell and take over its machinery, and then they're able to actually reproduce. They don't really metabolize their own until they attack a host cell. They're not made of cells either. They're very simple in their structure, made of just some sort of genetic material, their DNA or RNA, wrapped around a protein coat. So for our purposes, we're going to consider them non-living organisms. There's these organisms, they're not alive, they're non-living things, because once again, they don't check off those characteristics when it comes to life. So let's go ahead and start our PowerPoint. And the first thing I want to make sure you guys understand about viruses is their size. They are extremely small compared to other cells that we've studied. In this picture, notice here we have part of a eukaryotic cell, an animal cell. Notice here we have a bacteria, and then here we have our virus right here. And notice how the virus is a lot smaller than the actual animal cell, eukaryotic cell, and then even smaller than a bacteria cell. We can't see these with a microscope, like a light microscope. We would need an electron microscope to be able to see viruses. And guys, when it comes to viruses, they come in a lot of different shapes. They come in many shapes, and actually the shape of a virus, along with the receptors they're going to have, pretty much are going to determine what type of cell they're going to actually infect. For example, these are some of the most common types of shapes of viruses. This is the one we're mostly going to study, and it has one of the coolest shape, which is a phage. This is a phage, also known as a bacterial phage. That's an awesome shape there. That looks very mechanical-like. It looks like, if you've ever seen the show Jimmy Neutron, it looks like something out of that show or something out of the Matrix. It has some DNA, and then it has kind of this just protein coat that's in different shapes. This is an electron microscope picture, and notice how it pretty much looks like that virus. Now, because it has this shape, it mostly only actually attacks bacteria. This is specific to bacteria. They attack bacteria, like E. coli is one of the main bacteria that they end up attacking. We have spherical viruses, which kind of look like this. Once again, just DNA, a capsid, maybe an envelope, a lipid envelope with some receptors on it. These are usually specific for animal cells or eukaryotic cells. For example, influenza, which causes the flu, is a spherical virus. COVID-19, the coronavirus, is a spherical virus as well. We have the adrenovirus that kind of looks like, I don't know, like a tetrahedral kind of structure. These are usually specific for also animal cells. For example, these is what causes like strep throat. And then we have cylindrical viruses, which are mostly kind of infecting plants. Like this is a tobacco mosaic virus that is specific to attack tobacco plants. So we have many different shapes. The shape, along with the receptors they have, are going to determine what type of cell they're going to attack. Now, when it comes to viruses, the main thing I want to focus on is, okay, they're going to attack a cell. These virus particles, when they enter an organism or when they try to get into the cell's like a fortress. Their job is to penetrate that fortress, and once they do, their job is to reproduce. That's the first thing they want to do. They want to make just copies of them. Since they can't reproduce on their own, once they're able to attack a cell, enter a cell, they're going to try to reproduce. And there's kind of two main reproductive mechanisms when it comes to viruses. We have the lytic cycle and the lysogenic cycle. So let's go ahead and look at these, and we're going to start with the lytic cycle. And guys, when it comes to the lytic cycle, this is where that cell that it attacks, eventually it's going to die. It's going to die at the end. It's going to burst, pretty much explode, and it's going to come to its death, usually within a couple hours. And this picture here does an excellent job of telling you what happens in a lytic cycle. Notice there's pretty much 5 steps. So we have first, attachment. The virus attaches to the cell. In this case, it's a bacteria. Looks like an E. coli bacteria. Second stage, insertion. It inserts its DNA. And once the DNA enters, that's when it starts to take over all the material. It starts taking over the enzymes of that cell. It starts taking over the proteins. One of the first things it does is actually breaks apart the DNA of the actual cell it's attacking. It fragments the DNA, so now that cell can't really do anything of its own, and now the viral DNA takes over. It starts making copies of all of its components, makes copies of its DNA, it makes copies of its protein fibers, then it starts to assemble itself inside, and eventually it gets so full of viruses. We're talking 50 to 100 viruses that eventually it explodes, and now it releases 50, 100 new viruses that are now ready to do the same thing, infect another cell and just make a bunch more copies. So once again, this is the lytic cycle. Once again, the cell is going to die pretty much. That whole cell ends up dying. It ends up lysing, hence why we call it the lytic cycle. And once again, usually within hours of being infected. So I actually want to show you a video of the lytic cycle. So let me escape here and pull up that video. Let me just make sure the computer sound is on. Yes, it looks like it's on. Okay, let me make sure I open it with the correct player.
The Lytic cycle is a viral reproductive cycle in which a virus takes over all metabolic activities of a cell, replicates itself many times.
Okay, let me rewind from the beginning. Sorry, I want to make sure it's big enough for you guys to see.
The Lytic cycle is a viral reproductive cycle in which a virus takes over all metabolic activities of a cell, replicates itself many times, then destroys the host cell. A bacteriophage attaches to a bacterial host cell by recognizing and locking onto a specific receptor site on the surface of the host cell. The virus then injects its DNA into the host cell. The empty coat remains outside the cell. Inside the cell, the viral DNA breaks down the host cell's DNA. The virus then takes over the total metabolic activities of the host cell. By using the raw materials present in the cell, the viral DNA directs the production of new virus parts. The newly produced viral components are assembled into complete new virus particles. The host cell bursts open and releases 100 to 200 new virus particles. These new particles can begin another cycle by infecting nearby cells.
So once again, notice close to 200 just now get released, it killed the cell, and now they're ready to infect other cells. So once again, the lytic cycle. Let's recap those steps. You have attachment, then you have entry of its DNA, you have replication, assembly, and then finally lysis. You have release, you have lysis of the cell, and that's when the cell dies. So hopefully, guys, make sure you go back and maybe rewind this and play it again to make sure you have these steps down and knowing what's happening in each step. Okay, so let's go back to our PowerPoint. So once again, this is the lytic cycle, but there's another reproductive cycle when it comes to viruses. Sometimes they could reproduce in what we call a lysogenic cycle. Now, the big difference here, lysogenic cycle, the cell doesn't die right away. It doesn't end up killing the cell right away. It's going to replicate just the DNA, just the genome without destroying that host cell. Now, some of the steps are the same. You have attachment, you have entry. The viral DNA gets into the cell, but instead of taking over the cell, what it does, it hides its DNA into the DNA of the host cell. It incorporates its DNA with the bacterial DNA or whatever host cell that it ends up infecting. And we call this a prophage. So a prophage is when you get viral DNA mixed in with that host cell DNA. And here's the genius part of this. Well, eventually that cell is going to divide. And when it divides, it's going to have to do DNA replication. So it's going to replicate all its DNA. And when it replicates its own DNA, it's also going to replicate the viral DNA that's integrated in there. So now all the offspring of that cell, all the offspring of the offspring, all the offspring of the offspring, they're all going to have a copy of that viral DNA. And what could happen is at any point, that viral DNA could pop out. And if it pops out, boom, you're going to get the lytic cycle. You're going to have that whole cell eventually die. So let me show you a picture of what this kind of looks like. So here we have both of them. We're going to focus on the lysogenic. So notice we have attachment and we have entry. But then notice what happens here, lysogenic. Notice how this in blue is our viral DNA, in brown, that's our bacterial DNA. And notice it gets in there and it integrates itself. So now it's kind of like hiding in there, chilling out. Notice the cell's going to divide. It makes a copy of its chromosome and it makes a copy of the viral DNA. And now, once again, all these generations after this will have a copy of it. And what could happen, and this is where we don't know exactly why it does this. There's a lot of different factors. Could be something like a pH change, temperature change, but it could pop out. And if it pops out, boom, it could go into the lytic cycle, burst the cell, and now release another 200 viruses out there. So once again, this is the lysogenic cycle. So it doesn't necessarily kill the cell right away. An example of a very well-known virus that actually starts lysogenic is the virus that causes AIDS. I'm sure most of you know that AIDS is caused by a virus called HIV. When people get infected with HIV, it first starts usually in a lysogenic cycle. It likes to hide in the DNA of usually immune cells. And then for some individuals, they live their entire life with just HIV and they never get AIDS. But some individuals, for reasons that we still don't know exactly why it occurs, that HIV pops out and they start making copies. It starts destroying all those immune cells and it develops into the actual disease of AIDS. And that's why it's a hard disease to combat, because once again, they have no immune system. Usually it's not AIDS itself that kills the patient, but it's like a simple cold or a simple flu, because they don't have anything to fight off those infections. So once again, lysogenic and lytic cycle. So let me see, I want to show you a video that goes over the lysogenic cycle. So let me show you this video. Make sure that I open it with the right player.
In a lysogenic cycle, viral nucleic acid becomes part of the host cell's chromosome and is replicated with it. Eventually, the virus enters a lytic cycle and kills the host cells. A bacteriophage attaches to a bacterial host cell by locking onto a specific receptor site on the surface of the host cell. The virus then injects its DNA into the host cell. Once inside the cell, the viral DNA inserts itself into a specific site in the chromosome of the host cell. The viral DNA is then called a provirus. During cell reproduction, the host cell copies the provirus genes along with its own DNA. The provirus is inactive at this time. When the cell divides, both the host genes and the provirus genes are passed on to the two daughter cells. At any time, a provirus may leave the host chromosome and enter a lytic cycle. When this happens, the host cell's DNA is broken down and new virus parts are produced and assembled. Eventually, the cell ruptures and new virus particles are released.
Now, one thing I would want to make sure I go over, guys, notice how they call it a provirus. Now, there's a difference between a prophage and a provirus, and it's a very simple difference. A prophage usually refers to viral DNA that's integrating into a bacterial chromosome, pretty much a prokaryotic cell, where a provirus usually is intended for viral DNA that gets integrated into animal DNA, into a eukaryotic cell. For example, notice this cell is more round. It's not really bacterial. This would be more of a possible animal cell or some sort of eukaryotic cell, maybe a yeast cell. So once again, the main difference between provirus and prophage is pretty much the type of cells they're attacking. So prophage DNA of the virus gets integrated into bacterial DNA. A provirus, so once again, prophage is viral DNA into a bacterial cell, a prokaryotic cell, where a provirus, that viral DNA gets integrated into a eukaryotic cell. That's kind of the big difference between them. So once again, this was our lytic cycle. Let me go back to our PowerPoint. And actually, I think this is where I kind of want to stop for this first lecture. I want to keep it just short and sweet right here. This, once again, is going to be a major component of your future quizzes, knowing the difference between lytic and lysogenic cycles. So make sure you go back and review this. If you're still having trouble, make sure you reach out to me for any help that you might need through Edmodo, through email. Well, guys, I'm going to go ahead and say goodbye at this time. Guys, make sure you continue to focus on your work. If you need any help, once again, with distance learning, make sure you reach out to me, once again, via email or Edmodo. Well, guys, go ahead and have yourself an awesome day today.